Hello class, welcome back to Bio 105 Lab. Today we're going to be working on the lab Enzyme Kinetics 1. In Enzyme Kinetics 1, we're going to be looking at varying concentrations of an actual enzyme and looking at the effect that has on reaction rates. So we've probably already talked in lecture about this, but the general sort of equation for an enzyme catalyzed reaction, let's say, is as follows. You've got an enzyme, you've got a substrate, they're in solution, enzyme and the substrate bind together, and this forms the enzyme-substrate complex. Then the enzyme catalyzes a conversion of the substrate into another molecule or product, which we term the product. So, enzyme plus substrate, enzyme-substrate complex, substrate's converted to the product, it is released from the enzyme, and now you have an enzyme and a product. Here's a key point though, the enzyme is still there. In an enzymatically catalyzed reaction, the enzyme is not consumed. So it goes right back to the beginning, binds another molecule of substrate, and converts it to product.
Exactly.
Yeah, nailed it. So what we'll be doing today is we'll be varying the concentrations of our enzyme, and we'll be measuring the amount of product produced over time.
In the first experiment, in the second experiment, we're going to vary the amount of substrate, and we'll measure the amount of product produced.
And we'll see how that varies over time.
And then just a quick refresher, when you form this enzyme-substrate complex, that is what's going to lower the activation energy of the reaction. So if you have your free energy here on the graph, and this is time or progress of the reaction, you're going to have basically an energy barrier that needs to be overcome in order for the reaction to proceed.
No.
And what enzymes do is lower that energy barrier. So this is the reaction without an enzyme. This is the reaction profile with an enzyme, and you'll see that that activation energy is lowered.
Yeah. And oftentimes this is accomplished because the enzyme and substrate have a sort of lock and key where they fit into each other, and the enzyme can actually destabilize molecules or bonds. So this allows the reaction to proceed. We've talked about it briefly in lecture, but going from a high energy state to a low energy state is encouraged, it's favored. It should be spontaneous, except for this little barrier here where an energy input is required. The reaction can occur without an enzyme, but an enzyme lowers the amount of energy required to make it happen.
Exactly.
Yeah.
So the enzyme we're going to be looking at today is the enzyme that makes your potatoes go brown. So when you're getting ready for Thanksgiving dinner and you don't want your potatoes to look brown, that's the enzyme we're looking at today. It's called polyphenol oxidase, and because it makes a brown product, we can use spectrophotometry to measure the amount of product that is formed, which is the product that we're going to be determining. So what you would expect the results to look like is, if this is the amount, the concentration of the product, and this is the time of the reaction, as the reaction progresses, the amount of product is going to increase until you reach steady state. And then, in the first experiment, we're going to vary the amount of enzyme that we add. So, do you want to draw up what less enzyme is going to look like on that profile?
So remember, we've got the same amount of substrate in all of during the entire course of this first lab. So there's only so much product that can be produced. This would be the maximum amount of product that you can get. Right there. Once every molecule of substrate is converted to a molecule of product. But if you have more enzyme, the interaction or encounter between enzyme and substrate is more likely. So it occurs more rapidly, but still plateaus at the same level of product right there. And of course, less enzyme, slower reaction, still eventually reaching the maximum amount of product that can be produced from the amount of substrate that we have.
Yeah.
And I think that covers all the background that everybody needs.
Yeah, well, I just wanted to show what happens if you add less substrate, though.
Oh, that's a good question.
In the second part of the experiment, we're going to lower the concentration of the substrate. So then what will happen is the steady state that you reach. So if this is our steady state here, that is going to be lower when you lower the amount of substrate. So maybe when our reaction goes up, we're only going to reach a level over here. So it will be lower than the steady state with a higher concentration of substrate. Okay.
Makes sense to me.
Yeah.
Hopefully
So
it makes
should we
sense
get
to you.
started?
Cool. Yeah.
All right, so we weighed out potato already ahead of time. So we have 20 grams of potato here. And what we're going to do is actually just blend it in our nice little food processor here.
Yep,
Blender.
that's the plan.
Okay. So 20 grams of potato, and we need 20 mils of water, which we have here.
That's just distilled
Actually,
water?
200, sorry.
Yeah.
200 mils of water.
I don't think we'll need all that, but it's
There.
there.
Just
Right.
fire it up.
I think I'll mute this, hopefully, in the video. If not, cover your ears or mute your speakers.
Whew.
Wonderful. So what we're doing here is extracting that polyphenol oxidase enzyme. It was in the potato. That's why the potatoes turn brown. So we have blended up some potato, and there's enzyme in that jug right now.
So what we want to do now is make sure we don't have any chunks of potato that are going to interfere with our absorbance reaction. So we're just filtering it through cheesecloth.
Yeah, cheesecloth is a nice filter for getting large chunks of things out. It's definitely not like a sanitizing filter or anything like that, but probably couldn't even make a decent face mask out of it, quite frankly. There we go.
So perfect. There's our beautiful potato extract, and we'll maybe
smells
let
great.
it settle for a minute or two just to get some of the starch granules out.
Sounds good.
Right.
So we will be adding in our own source of substrate, which in this case is catechol. You can see on, well, in the middle of your of your protocol for today's lab, that catechol is what's actually converted into another molecule, giving it the brown color. And this reaction is catalyzed, as we've already talked about, by polyphenol oxidase. Good, great.
So catechol is the substrate. And because in the first reaction we're going to vary the amount of enzyme, we still want our reactions to have a constant volume, right? So in order to make up the extra volume and keep the volume of the reactions the same, we're going to add a little bit of inactive enzyme. And the way we're going to get inactive enzyme is actually we're going to boil some of the potato extract to inactivate or denature the enzyme.
Yeah.
Okay. So it's settled a little bit. You can see a nice kind of white precipitate at the bottom. So I'm going to pour out half of it.
And remember, in lecture we've talked already about protein structure and denaturation. Usually the primary forces that denature or destroy the structure of a protein are considered as heat and pH, at least in a Bio 105 course or something like that. So you can see that the enzyme or the extract already has a color. If we added less extract in order to vary the amount of enzyme, we would be diluting that color and we wouldn't be getting accurate measurements in the Spec 20. That's why Bridget right now is preparing a boiled and unboiled potato extract beaker.
Yeah. So you can see the starch granules at the bottom there. You see that white? We kind of wanted to get rid of that. So I'm going to go ahead and boil this.
Stupendous.
And then we'll get ready to set up the reaction.
Sounds good.
Okay.
Okay, so we have our boiled and unboiled enzyme, and now we are setting up our tubes as shown in your protocol under part 1. All right? So every tube is going to have 5,000, every tube is going to have a total volume in it of 6,500 microliters. All right? Volumes need to be constant. We need to have the same amount of total potato extract in each tube. But some are going to have boiled, which is inactivated enzyme, and some are going to have unboiled, which is containing an active version of that enzyme. So right now, Bridget is adding distilled water to all of our tubes in the volumes shown in column 1 of your table in the protocol, right here, using a nice little pipetter there. Fun device, uses suction to draw the fluid up. It's much better than the way they used to do it, which was mouth pipetting. So where they just literally put their lips on one of those things.
No mouth pipetting in the 105 lab.
No, even if you were here, we wouldn't make you do that. Great.
All right, so I have, in tube number 1, I have 5.5 milliliters or 5,500 microliters. And in all the others, I have 5,000 microliters. Okay, so they should all be even across. They look okay.
That looks good to me.
All right, so now I'm going to do the...
Yeah. So now Bridget is going to be adding in, would you like to start with the boiled or unboiled enzyme?
I'm going to start with the boiled.
Makes
it's
sense.
Just hanging out there.
Yeah, cool. So now Bridget is going to be adding varied amounts of the boiled enzyme. Once again, that's there so we're not diluting the natural color of the potato extract, which would interfere with spectrophotometric methods on account of the fact that the spectrophotometer measures essentially color. In other words, the amount of reflectance and absorbance of different wavelengths of light. If you're not comfortable with that concept, reach out to your TA or reach out to one of us and we'll talk about colors. It'll be fun. All right.
All right, and I'm using the P1000 to do this because I need to measure out between 500 and 1,000 microliters.
Yeah, and with it would be great to get you oriented to pipettes. I think we've talked about them in a couple of other labs. The micropipetter does say near the button at the top the range of volumes which it can effectively pipette. A P1000 can handle between 100 microliters and 1,000 microliters. In other words, a milliliter. So 1/10 of a milliliter to a milliliter. All right. Fantastic. Let's see. Bridget is making it through these tubes. We're also going to need to, do we blank in this one, Bridget?
Yes, we do.
We do blank. So what do we use to blank our tubes in this experiment, Bridget? Or blank our spectrophotometer?
Well, we first want to blank it with distilled water, because that's what the major part of our tube here is.
Yeah.
So the first we're going to blank it with distilled water, and then we're going to use tube 1.
Yeah.
Okay.
Makes sense to me.
All right. So I've added the boiled enzyme. Now I'm going to add the unboiled enzyme.
Yeah. So right now we're adding in, as Bridget said, the enzyme is being added in. There's going to be a small amount of substrate present within the potato extract, but we're going to be supplementing that with our catechol, or just an extracted version of the substrate. So you can already see actually that the unboiled enzyme compared to the boiled enzyme has a very different color. And that's because polyphenol oxidase is still operating in the unboiled enzyme or the unboiled extract.
And that's why we do tube 1. So tube 1 is really there to correct for any of the endogenous product that's forming in here.
Makes all the sense in the world, because you can see in tube 1 there is no catechol added. And that's why tube 1 sort of serves as our blank. The color change that is due to endogenous catechol should be more or less consistent between all of our tubes. At least we're hoping as much. Or at least all the tubes containing unboiled enzyme, I should say, unboiled extract. Fantastic.
Okay.
Is there anything else interesting about this? Well, spectrophotometer, right? We measure, the spectrophotometer gives you 2 different values. It gives you your absorbance value, which is how much of a particular wavelength of light is absorbed by your sample. And it gives you a transmittance value, or the amount of a particular wavelength of light which passes through our sample. Remember, we've talked about this before, but the spectrophotometer works by passing light through a sample of a known wavelength, then it measures the amount of that light which makes it through the sample. Stupendous.
All right, so we have all our tubes set up.
Great.
We've added water, we've added boiled enzyme, and we've got unboiled enzyme, and we have a total of 5,005. Well, we're going to have a total of 5,500 in all of them.
Or
once we add the
6,500, right?
Oh, okay.
Yeah,
Yes.
yeah.
Yeah. All right, so we're just going to add the catechol now. Because we're looking at kinetics here, we want to make sure that we keep the timing really consistent, all right? So the best way to do this is actually to stagger the reactions, because the spectrophotometers, you use a special little cuvette in here, and we have to transfer the reaction from this large tube where we've got lots of nice aeration, because this is an oxidation reaction, into the smaller cuvette so that we can measure the absorbance. And that takes time, right? So we have to be able to correct for that time. So we're going to stagger our time points so that we are not getting any discrepancies in our timing.
Makes sense to me.
Okay, so we've got a timer. I have a timer on here.
Got one there? Okay, perfect. Yeah, so in other words, every minute we will add a catechol to our first tube. That'll be time point 0. I'm sorry, actually there's no catechol in the first tube, but we'll be measuring that at time point 0. At minute 1, we will put catechol into tube 2. At minute 2, catechol goes into tube 3, etc., etc. This means that when we measure at minute 2 for tube 1, 1 minute later, it will be minute 2 for tube 3. It's a little clunky to get your head around, but hopefully that makes sense. You'll see us do it, and then it should be a little more understandable exactly what we are doing. Very good.
Okay, so I have this set at 500 for the catechol. Should we go ahead and measure tube 1?
We might as well. And can I have can I use that data sheet to enter in our information? As we go. Stupendous.
So just like in the carbohydrate lab, Jonathan's going to record the percent transmittance, and then you guys are going to work out the absorbance using that formula.
Yep, we will post up the raw data as we've been doing, and you're responsible for completing basically the lab handout. As should be expected. Now, the Spec 20 does make a little bit of noise when it's actually kicked on, so please bear with us. I'm going to try to muffle or edit out that sound as best I can, but it might be a little annoying buzz in the background. All right, let's crank this thing on. Oh, and could I please have a cuvette of distilled water with which to blank the machine. If you're interested in the specifics of what's going on behind the scenes here, please feel free to take a look online about Spec 20 operation. I'm sure that in one of these labs we'll have a nice sort of face-on video for the students.
All right, and so when you blank a spectrophotometer, you're actually setting the absorbance or the percent transmittance to 100. So what that's doing is it's allowing all the light to go through.
Yeah, and that tells us that the amount of light absorbed by just the distilled water, because we don't want to be measuring absorbance of the distilled water or transmittance.
All right, so tube number 1. So this is the endogenous potato. It's got no catechol in here. We're just seeing how much of the potato on its own is turning brown. I'm going to grab some wipes.
Fantastic. And yeah, as soon as we add the catechol in, we will begin our timer and then start our sequence of adding every minute.
Okay, whew, you ready?
This is pretty exciting. This is a stressful lab usually, so here we go. We got a nice 81% transmittance value in our endogenous.
Let me just do a little test here.
Might as well, yeah. We gotta get that dialed in.
There we go. I'm just checking to make sure it's not going to come out when I have it mixing. Okay, Jonathan.
Oops, sorry. Okay, here we go. Get this out for you.
Here.
And let's see. So you'll add the catechol, and then
I'm going to add the catechol, start the timer, and hand you the tube.
perfect.
I'll vortex it, then I'll hand you the tube, you pour it into the cuvette and measure.
Perfect.
Okay, teamwork.
Yeah, and that's just an essential step in this lab, is taking the minute to just make sure we all have a clear game plan.
And catechol is poison and it's toxic and it's nasty, so we're wearing gloves. Being careful. Give it a quick swirl.
Nice.
Yeah.
Perfect.
Okay, start the timer. Alright, and then when we get to exactly 1 minute, I'm going to add to tube number 2.
Okay. Value there. And this is usually a very stressful lab for people, but with our vast experience, a minute feels like ages. One fun tidbit about this lab is when we boil the enzyme or the potato extract, it does usually make the room smell like poorly prepared French fries.
Mm-hmm.
So
okay,
it's always fun. We're right there. Cool, cool, cool.
You're the dummy that don't believe in science. All your projects always be denying. You're the dummy that don't believe in science. All your projects always be denying. And I got thrown on half my words. I've got thrown on half my words. You're the dummy that don't believe in science. All your projects always be denying. You're the one I love, you're the one I wanna give to. Things got made in half the time. You're the one I wanna give to. Recognize that I can be here lately. You realize things can go away too. You're the dummy that don't believe in science. Written on your forehead, always be denying. You're the dummy that don't believe in science, written on your forehead.
Welcome back. So we found kind of what you would expect there, that unboiled enzymes, they are higher concentrations of unboiled enzymes are more rapidly going to decrease transmittance. And that means more product has been formed more rapidly. Total amount of product is going to be the same. So now, why don't we vary our substrate concentrations?
Okay.
So you'll see the raw data set once again. Take a look at your protocol to understand exactly what we're doing. But we're just going to go ahead and cut here, and then we'll get to it. All right.